The morphology of cells on microcarriers varies widely with cell strain and even with individual clones. Even for the same strain, the morphology may also differ when cells are cultivated on different types of microcarriers. Normal diploid fibroblasts, such as human foreskin cells FS4, readily spread out once they attach to microcarriers. However, subtle morphological differences can often be seen between controlled charge microcarriers and collagen or gelatin-coated microcarriers. In general, cells are much more elongated on collagen or gelatin-coated microcarriers than on controlled charge microcarriers. These cells exhibit typical social behavior of normal diploid cells and often grow in polarized fashion, as typically observed on Petri dishes. Confluence is readily achieved in a few days, and cells do not grow beyond the confluent monolayer stage as on microcarriers. The morphology of continuous epithelial cell lines such as Vero cells is very different from normal diploid cells. Cell growth on microcarriers often results in patches after 1 or 2 days. Given sufficient medium change, cells can grow to a very high concentration. The monolayer density, in terms of number of cells per unit surface area, is about 3 times that of FS4 cells. They can even form multilayers on microcarriers. Chinese hamster ovary cells also form multilayers on microcarriers. If sufficient medium change is still practiced after reaching confluence, they eventually form clumps. In relatively old cultures, clusters of cells may localize in some part of a microcarrier. Some cell types can be maintained on microcarriers for as long as 6 to 8 weeks. During that period, some microcarriers may have had cells fall off, while others still have clusters of cells on them. Or a number of microcarriers may form clumps,